our ductus epididymis, right? So epididymis, sorry, with the pseudostratified columnar epithelium, and these are the stereocilia. So we move on, and here we will have spermatic cord. So as you remember, spermatic cord is a structure which is going to be connecting your testis all the way to your prostatic urethra. And inside of the spermatic cord, you have lots and lots of structures. So here are the contents. And in your anatomy practice, you have to check if you have ID of the spermatic cord, because if you pick this one, there's a very particular way they want you to talk about. So you can't just randomly be like, this is testicular artery, this is vas deferens. You have to start, because it's going to be open like that, right? So you start from the most top and you move deeper and deeper, and then you come out again. And along your way, you have to show all of those things, because all of the contents that you can see in the spermatic cord, they're located in a very particular fashion. So one of them is lateral to another one of them is medial to another one. So they want you to kind of do that as well. So spermatic cord is a cord-like structure in males. Inside of it, there's a very important structure, which is the ductus deferens, right? So it's the most important thing. That's why we call it spermatic cord, because as you know, through the vas deferens, your spermatozoas are reaching the prostatic urethra. Spermatic cord is ensheathed into 3 layers of tissue. So the most inner layer, this is the internal spermatic fascia. Internal spermatic fascia arises from the transversalis fascia. Transversalis fascia, this is the fascia of your abdominal wall, right? So you remember this. So abdominal wall will have it there. Next layer in the spermatic cord, a bit outside, this is going to be cremasteric muscle and fascia. Both of them, muscle and fascia, are arising from the internal oblique muscle and its fascia as well. So of course, cremasteric muscle will come from internal oblique muscle of your abdominal body cavity and abdominal body wall, sorry. And cremasteric fascia will come from the internal oblique fascia. And the most outer will be the external spermatic fascia, and it arises from the aponeurosis of the external oblique muscle. So very important to remember that the only thing that does not give you anything in the spermatic cord, that's going to be transversalis muscle. Anybody knows why? Why transversalis muscle does not give you anything in the spermatic cord?
Because it's a worded reason. Because it doesn't, because the spermatic cord goes from inferior.
Yeah.
I don't have an actual reason, I have a theory, but it's not.
It's not inferior, just tell me what is it passing through? What is spermatic cord passing through?
Spermatic cord is passing through the abdominal cavity, it reaches the prostate, but it passes through the testicular artery. It has the testicular artery within it. It passes through
you're
near the
starting to blub enough. Okay, so this is going to be passing through the now I forgot it. It was so close to me. How do you call this canal? Inguinal canal. Okay, so it passes through the inguinal canal, and you have to know how to make inguinal canal. So what it consists of, and transversalis muscle within the area of the inguinal canal, it has a whole appearance. So it basically, transversalis muscle in a way makes the roof of the inguinal canal only. So when the spermatic cord passes through it, it passes through this opening in the transversalis muscle, so it doesn't get anything from it. The rest muscles, so internal and external oblique muscles, they have a bit of an area inside of the inguinal canal, so they are internally there. So when the spermatic cord passes, it gets ensheathed by these muscles. So the reason why transversalis muscle does nothing, because it actually forms this particular name structure. This is called the internal ring of the inguinal canal, and it's a ring-like structure, so there's an opening there. What can you find inside of the spermatic cord? You can read it all here. Okay, so in anatomy, you will also learn it. But basically, the most important thing for us is of course, the vas deferens. So here is the cross section of the spermatic cord. Okay, so vas deferens is here. Outside of it, you can see branches of the testicular artery. You can see some lymphatic vessels there. And you can see this venous plexus, which is called pampiniform venous plexus. Outside of it, you can see that there is some kind of skeletal muscle, and the skeletal muscle is going to be cremasteric muscle. Again, cremasteric muscle arises from the internal oblique muscle. This is a skeletal muscle, so cremasteric muscle will also be striated. It will also be skeletal muscle as well. So vas deferens, we talked about it. All of those things we talked last class, so make sure you refresh your memory there. Right, so you remember the most distal portion of it dilates and it is called ampulla. Epithelium is going to be the same as in the epididymis, but stereocilia will be shorter. They will not be as long as in the epididymis. And muscularis layer will contain 3 layers as well, very similar to your ureter, right? Very similar to your urinary bladder. So inner longitudinal, middle circular, and outer longitudinal layers. So these layers are also the same in your urinary bladder as well. So this is your vas deferens, right? So you can see pseudostratified columnar epithelium. These cells are principal cells. These cells are basal cells. And on the top of them, right here, you can see stereocilia. Of course, these stereocilia are nothing like the ones which we saw in the epididymis, right? In the epididymis, they were this long. A bit of the lamina propria under, right? So this area right here, this is lamina propria, and here are the 3 layers of the muscularis layer. Okay, so nothing interesting. Okay, pampiniform venous plexus. You remember what was its job?
To drain the testicular artery, unless I'm confusing it with a different.
This venous plexus will be wrapping around testicular artery. For which purpose? For cooling down the blood in the testicular artery, right? Because don't forget, your testes are outside. They want a 2 to 3 degrees lower temperature than the body, and testicular artery is coming from the body, so this is not good. So this is why pampiniform venous plexus will be wrapping around the testicular artery to cool it down. So here you go. These are veins. So these are pampiniform venous plexus. So does it look like a vein?
Not really.
Sorry, what? This?
No, really. The wall is very thick. Agree with me?
Yes,
So this
is a
not
thing?
your typical.
Wow.
Sorry?
So okay, yes. Because it was weird. I wouldn't have called it a vein.
Yeah, that's what I'm saying. So this is very, very weird. So these are veins. Okay, so take a look here right next to it. This is an artery, for example, right? So this is testicular artery. And this is the pampiniform venous plexus. It's a vein. So as you can see, this is a very, very weird vein. And you need to know that these pampiniform venous plexus, the veins that are in this venous plexus, are very special because unlike any other veins, they will contain additional muscle layer. So their tunica media is very special because it actually consists of 2 layers instead of 1, very similar to your arteries as well. So the reason why they're so thick, because they possess outer longitudinal layer of the muscles. So they have actually 2 layers there. They have the inner circular and the outer longitudinal layers within the tunica media. Could you tell me why? Why would that be the case? Why would they need this additional?
Because
layer?
they need to circle. They're looping around, so they would need more muscle.
So which way are they pumping?
Back towards
they're
the abdominal cavity.
up, and they're pumping against the gravity. Okay, they're pumping up and they're pumping against the gravity. Plus, as Alon have said, they're wrapping. Okay, so it's even double the distance of that you need to pump the blood from the testis to the body cavity. So that's why there is additional layer of muscles there. Okay, so there is additional layer of muscles for that purpose. So you can actually pump more efficiently because you're wrapping around testicular artery and you're pumping from down up. Okay, here we have right next to the pampiniform venous plexus, we have a cremasteric muscle. And even with this magnification, you can see that this muscle is actually skeletal because these are the bundles of the skeletal muscle fibers, right? So it's striated muscle. Where did this cremasteric muscle come from? Internal oblique muscle of the abdominal body cavity. There is something called cremasteric reflex, and cremasteric reflex has nothing to do with this cremasteric muscle, because you will see that testes, they will also have cremasteric muscle. In anatomy, you will see the scrotum, right? The sac which contains the testis will have cremasteric muscle there as well. And cremasteric reflex is when you take something sharp, like a pen, and you slide it from distal to proximally on the inner thigh. So you begin somewhere where the knee is and you start to slide this pen towards the scrotum. You will see that the scrotum will contract because danger, okay? So it's one of those cremasteric, it's one of those primitive reflexes that our bodies have. So cremasteric reflex is when the scrotum will contract and it will pull the testis closer to the body if there is anything touching inner thighs of the male, okay? Because that will, if there is something touching inner thighs, then what is stopping touching the testis, right? So, and this is dangerous. So this is why they kind of go back into the body. Seminal vesicles, we have talked about them so much last class, okay? So they are glands, right? So it's the area where we store additional semen, right? And yeah, we make fructose here. And don't forget, ejaculatory ducts is like a fusion of the duct of the seminal vesicle and ampulla. But we need to talk about something that they might ask you in anatomy. There is something called verumontanum. And verumontanum is present in the prostatic urethra. So when you open up the urethra, you will see two mountains, verumontanum, okay? You will see two little bulges within the prostatic urethra. So these verumontanum, they are bulges where the ejaculatory duct kind of protrudes through the prostate into the prostatic urethra. So I repeat, in anatomy, especially Schorzoli, he could ask you this kind of question about the verumontanum. These are just kind of bulges in the prostatic urethra through which ejaculatory ducts are opening up. So here's your seminal vesicle. You can see that it is highly convoluted. So technically it's one gland that is just kind of wrapping around several times. So you can see it here. So if we magnify, this is the epithelial protrusion, so mucosa. Here we have lamina propria, okay? So there is no muscle. So all of this is lamina propria. So it's a gland. Seminal vesicle is a gland. Epithelium consists of the columnar epithelium, okay? So it is columnar. Sometimes it's simple columnar, sometimes it is pseudostratified columnar. So it depends. The most important question here, what does the height of this mucosa depend on? Basically, what will increase the amount of mucosa in the seminal vesicle? A hormone of some sort? Which hormone?
GnRH, but probably a secondary effect of GnRH.
GnRH is just affecting pituitary gland, so it's
yes,
not
I know. So which one? Which
So of
one?
course it's going
LH.
to be testosterone. Okay, so of course it's going to be testosterone because LH will just simply go to the Leydig cell and Leydig cell will make testosterone. So don't forget there are two major hormones, dihydrotestosterone and testosterone. What does dihydrotestosterone work on?
Well, not this because this works on this. On the Y chromosome, Y gene expression. Well, not exactly, but
my question was, Alon, just calm down. My question was, what two glands are the DHT working on? Dihydrotestosterone. Not what does it do, just on which two glands is it working? Because testosterone is working on seminal vesicle.
Prostate.
Prostate is one of them, and another one
Epididymis.
is also considered to be a glandular tissue because it will make something. You will see that's going to be penis. Okay, so prostate and penis, these are the two areas which are under the effect of dihydrotestosterone. Everything else is under the effect of testosterone. So this epithelium, this mucosa, it will get longer, it will get much taller if there's more testosterone. So more testosterone, more seminal fluid, right? So more storage of the semen here. So yeah. There is another slide here. So this is also seminal vesicle, right? And you can see that right next to the seminal vesicle, there is some kind of structure here. What could it be? Right next to the seminal vesicle.
Prostate, urethra, no, prostate.
I would say testes, but it doesn't make sense. Seminal vesicles.
So don't say that because it's not testes.
No, it doesn't make sense. As I said,
okay,
seminal
so
vesicles, and they fuse with the ampulla, but the ampulla doesn't.
So what is this then? What is right next to it then? This is the ampulla of the ductus deferens. Okay, so this is the seminal vesicle, and this is the ampulla of the ductus deferens. You can see that the lumen starts to dilate. Okay, so it's a bit bigger structure. Okay, so we talked about verumontanum, you remember, right? So that's just like a protrusion of ejaculatory duct into the prostatic urethra. In English, verumontanum are called seminal colliculi. So definitely you should have heard these names, seminal colliculi. But Schorzoli, he likes the Latin, so verumontanum. This is prostate. Prostate is a gland. So here we have all of this epithelial tubular alveolar glands, which are making seminal fluid. And it is surrounded by the fibromuscular stroma. Okay, so this area right here, this is the fibromuscular stroma. So we magnify the same situation. These are the glands, prostatic glands that are making seminal fluid, which is alkaline fluid rich in prostaglandins, ascorbic acid, some proteins, etc. And this is the fibromuscular stroma. Inside of these glands, you can see this. What is this? We already saw the structure like this in the brain. This is prostatic sand. How did you call brain sand? Corpora arenacea. And it was present where?
It produces melatonin, not melatonin.
Yes,
melanin in the pineal gland.
Pineal gland, yes. Okay, so it's going to be in the pineal gland. So corpora arenacea is going to be in the pineal gland. This is in the prostate. So this is the prostatic sand, and it is called corpora amellacea, but it consists of exactly the same things. So it is calcium phosphate precipitation. So as a male, you have two areas where sand will be, and that's pineal gland and prostate. As a female, just one, so it's the pineal gland. So this is corpora amellacea, calcium phosphate precipitations. So we look at the prostate gland again, but then we use this staining, so Golder's trichrome. Golder's trichrome is very useful to show you smooth muscle cells. How many smooth muscle cells we have exactly in the stroma of the prostate gland. So again, all of these structures, these are your tubular alveolar glands, right? So nothing interesting. They're making prostatic fluid. There's a bit of the corpora amellacea in it located, sure. Everything else is the fibromuscular stroma. And if you look at it, right, so this is the fibromuscular stroma, you will see that it has lots and lots of these red cells, dark red, almost brown cells. Which cells are these?
Smooth muscle.
Smooth muscle cells. So what is this stadium trying to show you? It's trying to show you that fibromuscular stroma has a ratio towards smooth muscle cells. So you actually have more smooth muscle cells in the connective tissue stroma of the prostate than you have fibroblasts. So these are fibroblasts, just their nuclei, right? These are smooth muscle cells. So you can see that majority of the cells in the stroma are going to be smooth muscle cells. And we reach for the penis. Okay, so what is inside of the penis? So penis consists of two dorsal masses of erectile tissue. We can see them here. So this is mass number 1, mass number 2. They are called corpora cavernosa. So this is a cross section. This is corpus cavernosum number 1, corpus cavernosum number 2. Inside of the corpus cavernosum, you can see spaces, this darker area. These are called cavernous sinuses. So darker areas are cavernous sinuses. They get filled with blood during erection. So these cavernous sinuses get filled with blood during erection. Outside of cavernous sinuses, so pink tissue, right? So like this one, this is going to be trabecula, okay? So they are called trabecula. Trabecula consists of smooth muscle cells. So this is corpus cavernosum or corpora cavernosum because there are two of them, cavernosa. But what is inside of each corpus cavernosum? Two things, sinuses and trabecula. Which one gets filled with blood?
The cavern.
Sinuses.
Sinuses, okay, so cavernous sinuses, they get filled with blood. And which one have the smooth muscle cells?
Trabecula.
Trabecula, okay, so the one which you can see here, the pinkish area, these are the trabecula. A bit lower, you will find a spongy area, which is going to be known as the corpus spongiosum, right? So it's a bit lower part. Inside of the corpus spongiosum, you will find urethra. Surrounding three bodies together, so two corpora cavernosa and one corpus spongiosum, surrounding them, there will be a super dense connective tissue, which is the tunica albuginea. Okay, so tunica albuginea will be nicely surrounding all three of them because for some reason, this picture shows that only the two of them, but in our lecture, they say all three bodies are outside surrounded by the tunica albuginea. But between the two corpora cavernosa, there is a invagination of the tunica albuginea here. And this is known as the medial septum. So there is a bit of invagination of the tunica albuginea between the two corpora cavernosa. And this is known as the medial septum. So I repeat, this red line right here, this is going to be the tunica albuginea, the rest of it, right? So it's kind of outside all of the three bodies. But between the corpora cavernosa, you will see that there is medial septum as well. So we have three bodies in the penis. Which ones of them are erectile? Basically, which one of them are responsible for erection? It's only corpora cavernosa. Corpus spongiosum will never be filled with blood. Why? Because if it gets filled with blood, then urethra will be compressed and ejaculation will not happen. So this is why this medial septum is very important because it makes sure that only the corpora cavernosa kind of will get compressed with these blood vessels. So they make sure that corpus spongiosum is definitely not compressed by anything. So it stays open, it stays wide open, so ejaculation can actually take place. In the most distal part of the penis, you will see that there is a bit of a dilation here, which is called glans of the penis. And notice that inside of the glans of the penis, you only have corpus spongiosum. So there is no corpora cavernosa there behind the glans of the penis. Okay, so glans of the penis is basically your corpus spongiosum in a way. You will see it. Two fascias will be outside of the tunica albuginea. There will be superficial fascia and there will be deep fascia. Deep fascia is also known as Buck's fascia, B-U-C-K. Okay, so B-U-C-K, Buck's fascia. So deep fascia is also known as the Buck's fascia. Above these fascias, there's some kind of blood vessels. So for example, above superficial fascia, there is superficial dorsal vein and artery. Above deep fascia or Buck's fascia, there are deep dorsal arteries and deep dorsal veins. So these blood vessels, they'll always be above these fascias. And it's quite important, we'll talk about it. Okay, so basically here we're talking about that skin of the penis is very thin and it's quite loosely attached to the rest of the tissues. Okay, except for the glans area, because that's where the skin is definitely super tightly attached there. No adipose tissue could be found. Okay, so that's important to remember. And in the uncircumcised males, glans are going to be covered by a fold of a skin, which is going to be known as the prepuce. And prepuce on the inner part, which is facing the glans, it will resemble mucous membrane. It will look like you're looking at mucosa, okay, at the inner part of the prepuce. But outside of it is, of course, going to be skin. So epidermis, dermis, right, and all of this area. Notice no hypodermis because no adipose tissue. So it's a very special kind of skin. It just consists of two layers, epidermis and dermis. Inside of the prepuce and inside of the penis, you will have lots and lots of nerve fibers. You will have sympathetic, parasympathetic, somatic fibers, and they will all do a different kind of function. Okay, so yeah, so here we're talking about erection. I'm lost because technically we shouldn't talk about it because in physiology you talk about it, but let's just talk about it anyway, in case they ask you. So basically in the flaccid penis, flaccid means that it's not erected, okay? So it's just relaxed, it's hanging. You will see that inside of the corpora cavernosa, we have the trabecula, you remember? So if I go back, for example, this picture, these are the corpora cavernosa. Inside of them, there are these islands of smooth muscle, which we call trabecula. And in the flaccid penis, these trabecula are contracted. They're in the contractile state. They're not relaxed. So again, in the relaxed penis, these trabecula are contracted. If they are contracted, blood cannot flow into the sinus. So this cavernous sinus will be empty because these trabecula are contracted. So how does erection happen? Well, due to some kind of stimulation, could you tell me, would it be sympathetic or parasympathetic?
Parasympathetic.
Parasympathetic, very good. These smooth muscle cells will relax. So trabecula, they start to enlarge. They are relaxing. As they are relaxing, the blood starts to flow into the cavernous sinuses, right? So they start to get filled with the cavernous sinuses. And as the blood is filling this area, you can agree with me that these corpora cavernosa, they start to expand, right? So they start to expand like that. Just one problem, can the blood go out from these cavernous sinuses? It can't. Throughout the entire sexual intercourse, it cannot. That's why the penis is erected. Why it cannot? Well, the veins are going to be compressed. So your superficial and deep dorsal veins are compressed. Why? Because corpora cavernosa are expanding. The more blood flows towards the sinuses, the more corpora cavernosa expand. That means that these veins get compressed, so blood cannot leave corpora cavernosa, it stays there. So your penis is erected. But then as we approach the last stage of the sexual intercourse, the ejaculation, you're going to see that the innervation switches. So in order to achieve this climax, in order to achieve the orgasm, you're going to see that sympathetic fibers are going to come in, okay, and they're going to do what to your smooth muscle cells? They will ask them to contract, okay, so these smooth muscle cells will contract. As they contract, you're going to see that ejaculation happens. So basically the semen gets protruded out of the urethra, which is going to be here. And as this happens slowly, within a couple of minutes, not more than 5 minutes, the smooth muscle cells here stay contracted. As they stay contracted, that means that no more blood is flowing to corpora cavernosa sinuses. That means that slowly the pressure starts to go down. A little bit of pressure goes down, these veins start to open up because nothing is compressing them anymore. So blood starts to go away from these corpora cavernosa sinuses into the veins and being taken away from the penis. So you relax, okay? So you come down to your normal state. As you know, there is a drug called Viagra, right? So where we do it on purpose, a male that cannot sustain the erection takes Viagra. So one of the side effects of Viagra, actually one of the, I wouldn't say side effect, one of the most important factor of Viagra is that a male has to ejaculate. Doesn't matter if the sex happens or not, because lots of older males, they take Viagra on the dinner with the female, for example, and then they hope that something will go there, but nothing happens. So they were like, okay, I took Viagra, maybe in 6 hours, this will go down. It won't. One of the most important things about Viagra that the male has to ejaculate, because again, ejaculation is a bit of a part, it's partially under somatic innervation and partially under sympathetic innervation. So if sympathetic innervation does not come in, you don't ejaculate, this trabecular will never contract. They will stay relaxed. More blood comes in. Penis after 5 hours turns blue because there's so much blood comes into this area, right? So that's why typically male will come in after like 6 hours of erection. And as you can you imagine 6 hours of erection? How does the penis feels like? It's blue. You cannot touch it. The male cannot masturbate. Nothing can happen. Okay, so what should we do? There's a huge ass needle that you insert right into the corpora cavernosa, and you physically remove just a bit of blood to make sure that these veins open up so they can get the rest of the blood out. You're laughing, but you will see that that's actually quite common case. Okay, it's actually quite common case because males, they're like, okay, I did not get past the dinner, so what should I do now? Nothing. Okay, I'm just gonna hope that this erection goes away. But that's actually written in the prescription. Like when you open up the Viagra pack, you should always read what does it say there, and it's written there. After taking, ejaculation must happen because this Viagra will make sure that your penis will be erected forever. And there were cases where it actually became ischemic. Okay, so it actually, like, that's how dangerous it is. Okay, let's take a look at your penis. All the penises that we have here are embryonic. Okay, so it's not like adult penis, it's an embryonic penis, so remember this. Okay, so here we have our embryonic penis, H and E. So this is prepuce, right? So this is the skin which is overlying the glands of the penis. This is the glands of the penis, right? These are the corpora cavernosa. This is corpus spongiosum. What is inside of the corpus spongiosum?
Urethra.
Urethra. Okay, so this area right here, this is going to be urethra. This area right here, which is outside of the corpora cavernosa, this is going to be the tunica albuginea. Okay. So 2 corpora cavernosa, 1 corpus spongiosum. Inside of it, you can see the urethra. This is the tunica albuginea, and don't forget that this area of the tunica albuginea, which is kind of between the corpora cavernosa and happens to be also overlying the corpus spongiosum, you call it medial septum, right? So this is like a protrusion of it. Inside of the corpus cavernosum, you can see that there are some kind of spaces. What are these spaces?
Sinuses.
These are the cavernous sinuses. They get filled with blood during erection, and these tissues are
Trabecular.
they're the trabecular. They are with the smooth muscle cells, right? So they are contracted in the flaccid penis, and they are relaxed in the erected penis. Okay, so make sure you remember that. So take a look where we are. Okay, so this tissue right here, this is prepuce, right? And what is this? This is glans. And what do you see? You see that this is embryonic penis. So this prepuce is attached to the glans. So if I go back, here we are. You can see that the rest of it here is already detached, but here it is still attached. As you know, prepuce should move very freely, right? So it should be moving very freely over the glands, but here it is still tightly adhered. For example, here it is still tightly adhered. So how do we call it? This is called phimosis. And this particular phimosis, this particular fusion of the prepuce to the glands of the penis, we call it physiological phimosis. Why? Because this should be happening. So in the embryo, this should be happening. So in embryo, the prepuce should be attached to the glands of the penis. So if this is the prepuce, what is this? This is the epidermis, and this is
Dermis.
dermis. And then what is this? That's epidermis again, but that's going to be the epidermis of the glands of the penis, right? And you can see that this prepuce is attached to the epidermis of the glands of the penis. So for some reason, they really want you to know this, okay? So basically the layers here are epidermis, dermis, that's epidermis, dermis of the prepuce. And here we have the epidermis and then dermis of the glands, because it also has a bit of a skin on top of it, right? So I repeat, physiological phimosis, this is something that should be happening. Some children are born with a very, very tight foreskin at birth. So what moms and dads are told is that they need to retract it physically, okay? So in the first year of this male's child, right? So they ask them to actively remove the phimosis by pulling it down, and hopefully it will be fully retracted. If it does not resolve by the age of 5 years old, then there is a surgery where you actually actively kind of cut off this area. Okay, so take a look where we are. So this is the glands of the penis. Here we have a vessel of some sort. Don't be confused. This is actually going to be a deep dorsal vein. Okay, so this is a deep dorsal vein of the penis, of the glands of the penis. This wall here is looking a bit thick. It's okay, because that's embryo. We're still making vessels. Okay, so this is why it's a deep dorsal vein. Tunica albuginea, right, and 2 corpora cavernosa. Here we have another slide, and this is again for you, so you can kind of see what's happening. It's a bit older embryonic penis, just a bit more older. What are these?
Corpus cavernosa.
So corpora cavernosa, because there are 2 of them. This one is
Corpus spongiosum.
corpus spongiosum. Inside of it, you can see
Urethra.
urethra. Yes. What is this?
Tunica albuginea.
Tunica albuginea. This is
Median septum.
medial septum. You can see that there are some kind of vessels here, what will they be called? And this vessel as well.
The deep artery and vein.
Yes, deep dorsal artery and veins. All of them here, they're just branches of the structure. How do you call the connective tissue on the top of the deep dorsal vessels? You call it deep fascia, deep penile fascia, or another name?
Box fascia.
Box fascia. And then here you have a little tiny vessel.
Superficial dorsal.
Superficial dorsal, probably artery. And on the top of it, you will have
Superficial fascia.
superficial penile fascia. And this is your skin, right? So this is the epidermis with dermis here. Okay, we're in the corpora cavernosum, corpus cavernosum. And what is this?
Trabecular.
Venus.
Yes, so these are trabecular. These are the islands of the smooth muscle cells. And these are
Sinuses.
sinuses. You can see that there are some kind of vessels here. These are called deep penile vessels. Okay, so deep penile vessels. So inside of the corpora cavernosa, whatever blood vessels are here, you will call them deep penile blood vessels. We talked about this part, right? So this is your skin, epidermis, dermis. Here we have, I was wrong, superficial dorsal vein, superficial dorsal artery right next to it. So above it, what do we have here?
The foreskin.
Superficial penile
Superficial
fascia.
fascia.
Okay, so foreskin is not here because foreskin is on top of the glands, but we don't have glands here. Okay, so this section is a bit behind the glands. Glands was on this slide. We saw this is the glands and this is the foreskin on top of the glands, right? But here there is no glands, so there is no foreskin. You're a bit proximally to the body of the penis. Okay, so then how would you call this fascia?
Deep fascia.
Deep penile or box fascia, and here you have the deep dorsal blood vessels, right? So arteries and veins. Okay, and lastly on the slide we have the male urethra, the distal portion of it. Basically, in the human male, the urethra is about 20 centimeters, starting from the bladder, okay, so all the way to the tip of the penis, it's about 20 centimeters, and it opens up at the end of the penis into the external urethral meatus. So this opening in the head of the penis is called external urethral meatus. This is the common pathway for the urine and semen during ejaculation. There are 4 parts of the male urethra: intramural, prostatic, membranous, and spongy. Which one is inside of the penis? Spongy urethra. So of course, if I go back and I ask you which kind of urethra is here, you'll say spongy urethra because it is inside of the corpus spongiosum, right? So this is going to be spongy urethra. Where the prostate is, you will have prostatic urethra. Between them, where the base of the penis will be, you will have membranous, and that part of the urethra which begins inside of the bladder, you'll call it intramural. Okay, so that's your that's your spongy urethra, because what is it inside? This is corpus spongiosum. This corpus spongiosum has been stretched a bit, so that's why it looks like not corpus spongiosum. It looks like corpus cavernosum, right? So cavernous one with the cavernous sinuses and trabecular, but this is not, because you can see the urethra. So this is actually corpus spongiosum that has been stretched a bit. So how is the epithelium? Okay, so in the spongy urethra, you have pseudostratified columnar epithelium. So you don't have urothelium, you don't have transitional epithelium. Transitional epithelium stops at the prostatic urethra. So already when you're in the membranous urethra, it already changes, right, to the pseudostratified columnar epithelium. So this is your spongy urethra. Everything is great. The most important thing is here. Okay, so right here, this structure, like this one, and this structure is a duct of this one. Okay, so it's a duct of this one. So this is a gland. Through this duct, it opens up into the lumen of your spongy urethra. What is the structure? This is a periurethral gland. Another name for it is a gland of Litre. Okay, so periurethral gland or gland of Litre. These glands produce a colloid substance, which is very rich in gags. And the purpose of it to protect your urethra from the urine. Because guys, don't forget, transitional epithelium stops at the prostatic urethra. So technically, whatever urine you're passing, it could damage the epithelium there. So in the membranous and in the spongy urethra, outside of them, you will find glands of Litre. You will find them there. And the purpose for them is to make this mucus, which will be kind of coating your non-transitional epithelium and protecting it from urine that you're passing. Okay, so this is a gland and this is a duct of this gland. Make sure you remember. Okay, thank God. Male is over. So let's take a look at the, tell me, what are we looking at? We're looking at the female. Yes. Okay. So we're looking at the female. So it's the last part, but it has 90 slides here. So that's for today and for the next class, okay? Because female has way more important information than male. So first, a bit of the anatomy. I think you understand that the same as male, female consist of the internal and external genital structures, right? Internal female reproductive organs. This is going to be your ovary, right? So your fallopian tubes and your uterus. What is another name for fallopian tube? It's called uterine tube. Uterine tube, or what is another name? Very old name. Nobody uses that name anymore. Oviduct. 3 names talking about the same thing. Fallopian, uterine tube, or oviduct. Okay, so internal female organs are ovaries, fallopian tubes, uterus, and vagina could also be considered internal as well. External are the ones which you see outside, right? So this is the mons pubis, the 2 types of lips, majora and minora, clitoris, and a vestibule. Anybody knows what is vestibule?
An opening, well, literally an opening.
Vestibule is the space between the 2 minora. So without actually stretching things apart, it's very important in your practical exam as well. If they ask you to show vestibule, you don't like spread the lips like that and be like, that's the area. In its normal position, normal, how it is on the cadaver, you take your forceps and you just very gently show that where the minora are, right? So that's just the space between them. Don't spread them apart, okay? Hymen, right? Very important thing in some cultures, and external urethral orifice as well. Okay, basically your female reproductive organs, like your ovaries, uterine tube, uterus, they are always undergoing some kind of changes. And these changes are related to the neural activity and to the hormones. So constantly there is something happening in the female reproductive tract, and it is regulated by some kind of neurons or some kind of hormones. When do we start the menstrual cycle? It depends, as early as 9, as late as 14. On average, it's about 12. Well, closer to the 13 years old, okay, on average, if we averaged out, but it depends on your family. So always you will have menstrual period when your mom had it. So that's pretty much how it is, okay? So if your mom had it at 10, you will have it a bit earlier than 10. It's always a bit earlier. It's shifting just a bit. Menstrual cycle in medical language is called menarche, okay? So that's how it is, okay? So menstrual cycle, menstruation, they call it menarche. How long is menarche? Again, it depends, okay? Every girl has a different, but on average, it's about 28 days, right? So we always take 28 days as the average. How do you start counting the cycle, right? Sorry, I'm talking about menarche. Menarche is not 28 days. The cycle is 28 days, okay? So the ovarian cycle is 28 days, not menstruation, okay? So menstruation is not 28 days, it's the ovarian cycle. Menstruation is on average how many days? Maximum 5 out of this 28. It could be more, it could be less, it depends on you, but on average it's about 5 days of this 28. And how do we start counting the cycle? Well, first day of menstruation is the first day of your cycle, right? So day 1 of menstruation is the first day of your ovarian cycle. Of course, menstruation is not happening with us forever. Closer to the age of the 51 years old on average, the menstrual cycle becomes infrequent. So it slowly starts to be there and then not be, and then again be and then stop. And eventually it stops completely. And we call it menopause, or in Latin, this is actually called climacterium. That's why in simple English, they call it climax. Okay, so climax is the area where you have basically reached the point of your life, the climax of your life, where you can no longer make babies, right? So on average, it is 51 years old. So let's talk about the ovary. Ovaries have 2 functions, the same as your testes: gametogenesis and steroidogenesis. The gametes that the ovary is making, they are called oocytes. So that's why the process of making oocytes is called oogenesis. Mature gametes are called ova. So the oocyte which has definitely matured, you will call it ovum. The steroidogenesis is production of steroids, right? So production of the steroidal hormones. How do you call these hormones? Well, estrogen and progesterone. So these are the 2 steroidal hormones that are made by the ovary. What is the job of the estrogen and what is the job of the progesterone? Like, to help you remember, so you don't have to memorize everything, what does it mean, progesterone? Right?
That's progestation.
Exactly.
I mean, if I break it down.
Progestation. So it means that it is pro-embryo. So this hormone has nothing to do with you as a female. It has everything to do with embryo. So think about this kind of question. What is in the best interest of embryo? And this hormone will make sure that you have it. For example, what is in the best interest of embryo? To have a super, super nice endometrium, right? Where it will be implanting, as you remember from last semester. So progesterone will achieve that, okay? So it will be promoting the secretory changes of the endometrium. What is in the best interest of embryo? To make sure that mammary glands are prepared for lactation. So progesterone will also be doing that, right? Proliferating your mammary glands. You are, as a female, you're a female because of this hormone, estrogen. So everything which you have as a female is because of this one, right? I have internal and external sex characteristics because of estrogen. Ovarian structure. So in the nulliparous female, so nulliparous means the female that have not born any children yet. Okay, so nullipara, it's the 0 birth, right? So they haven't given birth to any children yet. The ovaries are paired, almond-shaped, pinkish-white structures, measuring about these numbers which they want from you in the anatomy room. Okay, so you need to remember it's 3 centimeters in length, 1.5 in width, and 1 in thickness. Here we're talking about anatomy, which you're going to read at home, because why waste time? Okay, so just there's nothing of importance in histo whatsoever, just anatomical structures, what is it attached to, which ligament, what runs in that ligament, and etc. Histologically, it's very important to remember that on the surface of the ovary, you will have single layer of cuboidal epithelium. So surface of the ovary is covered by simple cuboidal epithelium. Some parts squamous, sure, but majority simple cuboidal. And this simple cuboidal epithelium is called germinal epithelium. In embryo, you should have learned last semester that they called it germinal epithelium because they thought that PGCs will be arising there. They thought that male PGCs and female PGCs could not be possibly arising from the same spot, because back in the day, they thought that male is more superior and female is more inferior, right? So how could the two genders have the same areas for PGCs? Of course, that's not the case. So that's why they thought that PGCs will arise from the germinal epithelium, from the surface epithelium of the ovary. And male PGCs will come from epiblast and blah, blah, blah. And then they later realized that that's not how it is, that it doesn't matter. PGCs will always come from epiblast, doesn't matter male or female. But the name kind of stuck. So they still call it germinal epithelium, even though it does not have any PGCs. Could you guys remember? Could you remind me, sorry, what are PGCs? Yeah, I figured by your faces when I say PGCs, everyone was like, what the fuck is PGC? Okay, but yeah, okay, so PGC is a diploid cell that gives rise to your gamete. It gives rise to spermatozoas or oocyte.
Oh, that.
So, yeah,
Hindsight
okay, so
bias.
if
Yes,
I have a
hindsight bias. Never
PGC,
mind.
so if I have a PGC inside of which I have XX chromosome, what will this PGC give me?
Female.
No, it will not give you a female because PGCs give you oocyte or spermatozoas.
Oh,
So
sorry.
how can it give me a female? It cannot give me a female. So it can give me either spermatozoa or oocyte. So which one?
Oocyte.
Oocyte. Okay, so it is going to give you an oocyte. This diploid cell carrying XX chromosome is going to split. It divides. And as it divides, you now have two cells. And what is inside of these two cells? Only one chromosome, which is X. Agree with me, 100% of the cells carry X chromosome. All of them will have X chromosome. So which cells are we talking about? We're talking about oocytes, because oocytes only have X chromosome. In males, we had a similar situation. So we also had PGC there. And also that PGC was also splitting into two cells. But which kind of chromosomes does PGC carry in males?
X and Y.
X and Y. So when the cell splits, you see that some cells have X chromosomes, some cells have Y chromosomes. In males, spermatozoas, 50 carry X, 50% carry Y. So you understand why now? Because PGC, that's why, because it is diploid cells. So when it splits, it gives you different kind of cells. Okay, so where does PGC come from in embryo?
Was the invagination in the 4th week?
I just said it right now, like about 2 minutes ago. I said it.
Epiblast.
Epiblast, very good. Okay, so primordial germ cells, they will arise from epiblast, both male and female. It doesn't matter, but they will both come from epiblast and give rise, go to the gonads, and in the gonads, they will give you either spermatozoa or oocyte. What does that depend on? It depends on the chromosomes that you're carrying, right? So if I'm carrying XX, that means that in the gonad, they will become oocyte. If I'm carrying XY, that means that these PGCs in the gonad will become spermatozoas. Histologically, ovary is composed of cortex and medulla. And in the medulla, you don't have any follicles. Could somebody refresh my memory? What is a follicle? Last semester we learned it.
Pure definition I don't have, but it's the...
Just what does it consist of?
It consists of the oocyte surrounded by the corpus luteum. Well, not corpus luteum.
Hold on. It's the oocyte, I agree. So in the center of the follicle, you have oocyte.
And then you have the layer which surrounds it.
Why is it called follicle?
It's the oocyte
surrounded by the follicular cells. Very good. It's the oocyte which is surrounded by the follicular cells. And these follicular cells will be changing their shape throughout the entire cycle, right? So at first, as you remember, they were squamous, then they became cuboidal, then became stratified cuboidal, and blah, blah, blah. Have I told you that this SCT will be embryo? Have I told you last class? Have I told you a class before that? So you can see that's true. So everything which you have learned last semester embryo will be asked here in this self-control test. If you pick the slide in the oral, you're not talking about histo. Nobody cares about histo. I point and that's it. You will be talking about embryo. How did it actually arise from? So that's why embryo 1 is very, very crucial. These past papers that you have done last semester will be now asked to see that do you understand what have you learned last semester. So make sure that your embryo 1 is very strong. So this is a follicle, and follicle, I repeat, consists of the oocyte, which is surrounded by the follicular cells. Okay, and where are these follicles located? Not in the medulla, in the cortex. This is very important to remember that you will not find a single follicle in the medulla. You will find all of them in the cortex. And these follicles are embedded in the super nicely made connective tissue, so stroma. And very important to remember that this stroma will also have smooth muscle cells as well. So you will see it on the slide that this is a very, very special stroma. It doesn't only have fibroblast and collagen fibers. You also see smooth muscle cells as well. So cortex medulla, where are the follicles?
The follicle
in the cortex, what is a follicle?
Cortex.
What is a follicle?
Oocytes around
Follicular cells in the ovary within.
It's the oocyte surrounded by the follicle.
Sorry, the oocyte within.
Okay, so we continue. So here we're just talking about that what is a follicle, that it is basically an oocyte surrounded by the follicular cells, right? All of them are distributed in the stroma of the cortex, sure. During puberty, you're going to see that all of these follicles are starting to grow. We'll see it right now. They're expanding, they're changing the way they look like. So by the by the middle of the cycle, you can make only one mature follicle. And this one mature follicle will rupture. And as it ruptures, it gives you this definite oocyte, which will be fertilized by the spermatozoa. Right, so that's what we saw in the embryology previously. Here it's just some numbers. So let's kind of refresh our embryo here. When did you start making follicles? When did you start making follicles?
Embryonic.
As soon as, of course, but which number? Like when exactly in the embryonic development? 5th week. Super early. Okay, you start making follicles 5th week of development. When have you finished making follicles? 5th month. Okay, so this is when you already have there. In the embryo, it says 7th month, but I will tell you what is happening in a second. 5th month, all of the oocytes are ready. You have made primary oocytes, they are all ready. And if you were to count how many primary oocytes have you actually made, you will count 7 million primary oocytes. So the first number that I want you to remember is that when you reach 5th month of development of the fetal life, you have 7 million primary oocytes. That's 7 million potential human beings. Okay? I don't know how. I know Israel is 10 million.
Population?
Yeah.
9 million.
Yeah. I know that Russia is 135 million. Okay. 7 million in one girl's oocyte, ovaries. Okay, so can you imagine? So agree with me, that's too many. Am I right? 7 million primary oocytes, that's too many. 7 million potential human beings. It's too much. So what should you do? You should apoptosis, okay? You should just remove them, remove majority of them. So how many are you actually born with? Out of those 7 million, you're born with 600 to 800,000. So majority of them are killed. In histo, they wrote 5 million. In embryo, it says 7 million. So that's why you're going to stick with what the embryo says. Okay, so 7 million in fetus are reduced to less than 20%, okay, of that number. So about 600 to 800,000. Okay, so that's how much. How many do you have when you reach the puberty? What does the embryo book says? Because that's how many when you are at birth. But when you start the puberty, how many do you have?
Well,
in embryo, they said 40,000. Okay, so in embryology book, they say 40,000. But honestly, this number, it could not be true. And it is not true. When you come to the 4th year OBGYN, the number is more like 240,000. Because if it was 40,000, you will reach menopause when you're like 29. Okay, so yeah, it's not possible. So it's more like 240,000. Okay, that's in real life. But again, disregard my 2 because you're still 2nd year medical students. If the embryo book says 40,000, we're going with that number. Okay, so 40,000 out of this one we have. Every month that you start the puberty with, every month out of these 40,000, how many do you choose?
1.
No, that's how many mature, but how many do you choose? I have to have a choice, an option. 15 to 20 every month, 15 to 20 follicles out of this 40,000 are being used, and only one of them will mature. So can you see how much waste is happening? We started with 7 million when you were a 5th month old embryo, to 800,000 when you were born, to 40,000 when you start the puberty. And even out of this 40,000, every month you still be choosing 15 to 20, but only one of them will mature. So what will happen to the rest of them? They'll just be dying, right? If I only choose 1 out of 15, what will happen to the rest 14? They cannot go back to the ovary and still be frozen and something like this. No, they will also undergo apoptosis. So that's why menopause is inevitable. Okay, you will definitely reach it at some time when all of these follicles will finish. When you finished all of them, that's when the menopause is. Okay, speaking of follicles, there are definitely different types of follicles. We have primordial, growing, and mature follicles. Primordial follicle is the one that you have made in embryo. That's why we call it primordial follicle. And in your ovary right now, as a female, in your ovary, majority of your follicles are primordial. So they are just sleeping. They're just waiting to be woke up. Okay, so to be woken up. Primordial follicles, due to some kind of stimulation, will become growing follicles, which are your primary and secondary, right? So primordial will become primary, primary will become secondary follicle. Secondary follicle will become tertiary or mature follicle, and mature follicle will become Graafian follicle. Graafian follicle is the one that you have at the end. Graafian follicle is the most mature version of your follicle. Could you tell me what stimulates this process, this kind of
LH spike?
No, LH spike is when this Graafian follicle will burst.
It's released,
and
sorry. Yes.
That's the LH spike.
FSH.
Of course. They are follicles, so they are stimulated by
Follicular stimulating hormone.
So the reason why primordial follicle becomes primary is because of the FSH, right? So all of these follicles, they should have some kind of receptors, and these receptors are going to be specifically for the FSH. The more FSH come in, right, the more I am growing up. So you remember how many do we choose every month?
15.
15 to 20.
15 to 20. But how many of them will reach the final Graafian follicle stage? Only?
Uno más.
Only one. So how do we establish that? That only one of them reaches Graafian follicle. Again, it has something to do with your FSH receptors. So the one which is reaching Graafian follicle stage
Will be the one which expresses the most FSH, doesn't it?
Exactly. It's the one which has the most of the FSH receptors. So if it has the most of FSH receptors, FSH comes in specifically to this kind of follicle and stimulates it to grow much faster. So it reaches the stage much faster than the rest of the follicles. Okay, primordial follicle. When do they start to appear? We start making them at 5th week of development, but you actually start to see them at 3rd month. You can see like individual primordial follicles at 3rd month. So I repeat, we start making them, we start the process at 5th week, but you actually see individual follicles at the 3rd month of the fetal development. What does the primordial follicle consist of? It consists of the oocyte surrounded by the follicular cells. What is the shape of these follicular cells? They are squamous. Simple squamous. So in the primordial follicle, the follicular cells are simple squamous. Could you tell me which oocyte is sitting here?
Primary.
Primary oocyte, very good. And primary oocyte, how many chromosomes does it have? Is it diploid or is it haploid?
Primary is diploid.
Primary is diploid because it is entering into the meiosis. It's entering into the meiosis 1 to divide the numbers and become haploid. So secondary oocyte is haploid, but primary oocyte is diploid. So the cell here is diploid cell. So this is the nucleus, this is the cytoplasm of the primary oocyte. Right, so I repeat, primordial follicle, this is a primary oocyte, which is surrounded by the squamous follicular cells. Okay, next thing that you're going to see that all of these primordial follicles are typically located in the most outer part of the cortex, just beneath the tunica albuginea. As you remember, this is a capsule which is outside of your gonads, testes, and ovaries. So most outer part of the cortex will have the most of the primordial follicles. You will actually see it there. The oocyte inside of this follicle is relatively small. It's only 30 micrometers. So you need to remember this. It has a very large eccentric nucleus, okay? And cytoplasm is referred to the ooplasm, contains sometimes Balbiani body. Have they ever talked about this body? Never, but I still give it to you just in case. If you have like Professor Rosa and she's in a bad mood, she could possibly ask you, but she never does, but just in case. Balbiani body, it's a very special body in the oocyte. By the way, this could be important for you next semester pathology. So remember this moment right now, okay? So because there is something, could be something wrong here. So Balbiani body is a structure inside of the cytoplasm of the oocyte. And you can think a bit of it as a bucket which contains different organelles. So it's like an organelle inside of which you have another organelles. It's like this little tiny, I don't know, like a bucket, like a box which is filled with other organelles. Which kind of other organelles? Golgi, mitochondria, lysosomes, and endoplasmic reticulum. Do you understand why would I put them into Balbiani body?
Repository, a storage.
But why? Because fertilization will be happening in the cytoplasm of the oocyte, right? So the head of spermatozoa will squeeze in here, and then the two nuclei will fuse together. That's your fertilization, right? So that's when the two nuclei fuse together, you have an end result called zygote. Take a look, the mitochondria here are very big. So that's why you put them all together into one part to make sure that you have lots and lots of space for fertilization to actually happen. So when the head of spermatozoa comes in, it doesn't bump into like lysosome or mitochondria or something like this, okay? So you kind of clear the area for the future fertilization to happen. So this Balbiani body is like a storage exactly of these organelles to make sure that there is available space there. Annulate lamella are not important, okay? So this is about them as well. It's not important. They never talk about them, so you don't need to know this. Your primordial follicle will have receptors for FSH. And when the FSH comes in, right, it binds to this FSH receptor and causes your primordial follicle to grow. So when primordial follicle grows, it becomes primary follicle. What is the major difference between primordial and primary follicle? The flat squamous cells that were follicular cells are no longer flat. Now they're cuboidal, right? So they become simple cuboidal epithelium. So follicular cells have changed their shape. So this is called primary follicle. At first, at this stage, you're going to see that this is like the first stage of this primary follicle stage. And here the epithelium is still simple cuboidal. And then the more time passes, the more cuboidal cells will start to appear, and your primary follicle enters into the second stage of its development, which is like a multi-layered stage. So these simple cuboidal follicular cells are now starting to proliferate. So are they single? Of course, no. Now they become stratified cuboidal follicular cells. So these stratified cuboidal follicular cells change their name. Now they are going to be called granulosa cells. So again, primary follicle has two stages. You will see two stages. In one stage, there is simple cuboidal epithelium. And another stage, it's stratified cuboidal epithelium. So which cells are here?
Simple,
How do you call them at first? At first, how do you call them?
simple cuboidal. Well, they are simple cuboidal. We call them follicular.
Follicular cells. So in the first stage of the primary follicle, it's just simple cuboidal follicular cells. So they were squamous follicular cells, but they became simple cuboidal follicular cells. In the late stage of the primary follicle, follicular cells change to granulosa cells. And what are granulosa cells, basically?
Stratified cuboidal
Follicular cells, very good. It's basically stratified cuboidal follicular cells. Stratified cuboidal follicular cells make something. They secrete a glycoprotein, and this glycoprotein is coating your oocyte here. And this glycoprotein is called zona pellucida. Okay, so zona pellucida is made in the primary follicle. It is made by the granulosa cells. So stratified cuboidal follicular cells will make zona pellucida. Zona pellucida consists of the three glycoproteins. You name them ZP1, ZP2, and ZP3. Out of three, I guess ZP3 is the most important one because it actually induces acrosome reaction. And what is acrosome reaction?
Decapitation. After decapitation of the head of the spermatozoa, you have the destruction of zona reticulum.
Exactly. So
yeah, let me simplify. I'm trying to simplify this.
Yes, you need to shorten your sentences. In the exam period, especially if you go towards the end, the professors have zero. It's so bad, and I'm really like, I don't mean to scare you all, guys, but the closer to the end of the exam period, the less patience professors have. Okay, so if you go in the first 3 weeks, they're very lenient. They will listen to you blabbering forever. Okay, they will be listening and they'll be very understanding.
Acrosomal reaction is the fusion of spermatozoa.
Can you imagine how many students are passing through them? It's almost 400 students are passing through them. So that's why by the time you reach the end of the exam period, like you're going 6th, 7th week, they will not listen to you blabbering. So for example, let's take the situation with Alon here. The question was, right? So what was the question? What is acrosome reaction? Alon starts to talk not about acrosome reaction. He starts to talk about capacitation reaction. He says it's the reaction which is after capacitation reaction, but that was not the question. And from personal experience and through 5 years of tutoring, guys, if they ask you a question, answer the question. Don't
Straight
start
up,
talking
yes.
about other things. So acrosome reaction, he eventually got to the answer. He said, that's the part where zona pellucida is being digested. But you should have just make your answers very short and very on point. Okay, so acrosome reaction is when zona pellucida is being digested. So as you remember, we talked about it in the last class. Spermatozoa possesses digestive enzymes, and these digestive enzymes are behind the acrosome. They're hidden by this protective layer. Capacitation, right, is when you remove the acrosome. But acrosome reaction is when these digestive enzymes are getting exposed. Are they active? No, these enzymes are not active. Something needs to activate them. And what is that something? ZP3 in the zona pellucida. So basically, zona pellucida activates its own digestion. It activates the digestive enzymes to do the work. So here we have still primary oocyte. Notice in the primary follicle inside still primary oocyte. So nucleus and cytoplasm. So we have talked about granulosa cells. This is very important. How do you call them? Follicular cells, which are becoming the granulosa cells, they also have some kind of junctions between them. As you remember, Sertoli cells, they had tight junction between them, and we call this blood-testis barrier, right? We called it Sertoli barrier. Between the follicular cells, there aren't any tight junction. Instead, you will see gap junctions, highly tested. So which junctions are between the Sertoli cells? Tight junctions, so that's why there is blood-testis barrier. Which junctions are between the follicular cells? Gap junctions. So there is no such a thing as blood follicle barrier. It's absent. There aren't any tight junctions, so there is no blood follicle barrier there as a result. Okay, connective tissue right outside of the primary follicle has a name, and this is known as the theca folliculi. So if I go back, right, so this area, which is outside of your primary follicle, this is known as the theca folliculi. So this is not theca folliculi because it is not outside of your primary follicle. Just the connective tissue, which is within the proximity of your follicle, you will call it theca folliculi. Is it clear? Right, so this is the theca folliculi. Theca folliculi later will differentiate into two layers, into the theca interna and theca externa. Theca externa is just connective tissue. It contains smooth muscle cells, nothing interesting. Theca interna, the inner layer, later will differentiate into the theca interna cells. And theca interna cells have lots and lots of LH receptors. And if theca interna cells have lots and lots of LH receptors, that means that they will be using available LH for something. For what? To make a precursor of estrogen. They will not make estrogen. They will make precursor of estrogen. Who will make the estrogen? Well, who is under the theca interna? Follicular cells. Follicular cells will have an enzyme called aromatase. And follicular cells will convert this precursor into the true form, which is estrogen. So let's do it again. How do you call the tissue outside of the primary follicle?
Theca folliculi.
Theca folliculi. Right now, this is just theca folliculi. But at the later stages, like in the secondary follicle or in the tertiary follicle, this theca folliculi will differentiate into two layers. How do you call these two layers?
Interna.
Interna and externa. Externa is, of course, the one which is outside. So it will just have smooth muscle cells, nothing interesting. But what will the theca interna have? The theca interna cells, and these cells have a receptor for LH. Okay, so theca interna cells will have a receptor for LH. If LH binds to theca interna cells, what will they make?
Estrogen.
Estrogen.
No, they will make precursor
precursor
of
of
estrogen. They will not make estrogen. They'll make precursor of estrogen. In physiology next year, next semester, you will learn the name. It's called androestradiol, something like this. Androestradiol. This is the precursor. What will happen to this precursor?
It will
Who
become
will
aromatic.
take it up? Which cells are below theca internal cells?
Follicular.
Follicular cells. Follicular cells will take up this precursor estrogen and convert it to
Aromatase.
estrogen.
Estrogen
Thanks
through
to
aromatase.
which kind of enzyme?
Through aromatase. Aromatase is the enzyme.
Aromatase. So which cells have LH receptor?
Theca internal.
Theca internal cells. And they will use this LH receptor to make what?
Precursor of estrogen.
Precursor.
Precursor of estrogen. Who will take up this precursor of estrogen, Jordan?
Follicular cell.
That's not a Jordan, but Jordan was very
Follicular
lucky. And because he's not listening, he's just staring, but he's not listening. He's thinking of something else. So follicular cells will take up this precursor of estrogen. And what do follicular cells have? Anybody else can answer because Jordan doesn't know.
Receptor for LH.
No, we're talking about follicular cells. We're done with the theca
Sorry.
internal
No,
cells.
no, no. Follicular
Aromatase.
cells.
They
No.
have aromatase, very good. Follicular cells have aromatase.
Okay.
And because they have aromatase, they will convert this precursor of estrogen into
Estrogen.
estrogen.
proper.
So a question, who makes estrogen?
Follicular cells.
Follicular cells, very good, follicular cells. Theca internal cells cannot make estrogen, they make precursor of estrogen. That's not estrogen.
Julia, I have a question.
Mm.
At this stage, you still call it follicular cells, not granulosa cells?
Very good. You technically call it granulosa cells. You're right. You're 300% right. You should call it granulosa cell, but I am saying it follicular cells, so I don't even know why I'm saying follicular cell. In my brain, to make it easier for you, because you already probably forgot what is granulosa cells, but you're 300% right. Technically, the proper way to say is absolutely that granulosa cells will make the estrogen, not the follicular cell. But you remember that granulosa cells are stratified cuboidal follicular cells. So at the end of the day, they're still follicular cells. But a more technically appealing would be if you say granulosa cell. That's 300%, yeah.
Okay, thank you.
Okay. Okay, so theca internal, theca external. Okay, so which kind of follicle is this and why?
Primordial.
It's primordial follicle, very good, because here we have a nucleus. This is the cytoplasm of the primary oocyte. And what are these cells?
Simple squamous.
Follicular cells, but their shape is going to be the simple squamous. So this is definitely primordial follicle. What is the location of primordial follicle typically?
Cortex of the oocyte.
Amazing. Cortex of the ovary, most outer part of the cortex as well, right? So right under tunica albuginea. So here, which follicle is this?
Yes.
Primary follicle, because what is the shape of the follicular cells?
Cuboid.
Cuboidal. And this is simple cuboidal epithelium. So technically, this stage is the first stage of the primary follicle, right? Because don't forget, the second stage is when these simple cuboidal follicular cells become stratified cuboidal follicular cells. Okay, secondary follicle. So my primary follicle should become secondary follicle. It is considered to be called secondary follicle only when this space appears. The space is called antrum. As long as you see a little tiny space here, this is called antrum. Inside of the antrum, there is a fluid. This fluid is called liquor folliculi. Liquor folliculi is made by the granulosa cells, right? So by the cells which is outside of this antrum. And who tells them to make this? LH. Okay, so LH will be responsible for secretion of this liquor folliculi into the antrum. So just a sec, guys. Sorry, guys. So antrum, instead of the antrum, we have a liquor folliculi. Liquor folliculi is going to be made by the granulosa cells. Granulosa cells are stratified cuboidal follicular cells, and they make this liquor antrum because we have LH, right? So LH will be binding here, and as a result, you will see it there, this liquor folliculi. These are the factors which are responsible for active growth of the secondary follicle. So as you can see, FSH is still there, so it still plays a very important role, but also calcium, and also for you, insulin-like growth factor. That's the one that they asked, for example, last year. Okay, so insulin-like growth factor actively participates in the growth of the secondary follicle. Oocyte maturation inhibitor factor. Who remembers this?
Is it OMI?
It's OMI, yeah, but what's its job?
To inhibit the oocyte from maturation?
Be more specific, because that's what it means. Oocyte maturation inhibitor.
Well,
so
I mean
can get this
division.
information from this, huh?
From division.
That's what maturation is. So you have to be more specific. Oocyte maturation inhibitor is made by the follicular cells. Follicular cells make it all the way here in the primordial follicle. Okay, so they are making OMI.
Oh, wait a second.
And
Is that
the person, too late. And
Yes.
The reason for this OMI is going to be is that your primary oocyte does not become secondary oocyte. You have to be way more specific. This OMI keeps your primary oocyte in a very specific stage. It keeps it in the diplotin stage. Diplotin stage is a sub-phase of the meiosis 1. Okay, there's a bit of a detailed stages there in meiosis 1. They divide it into even more. So not 4 stages in meiosis 1, there are actually 8 stages there, according to genetics. And in one of them, the primary oocyte is kept. That's the diplotin stage. So diplotin stage in the meiosis 1 is a resting stage for the primary oocyte. And the reason it is in this stage is thanks to the OMI made by the follicular cells. Okay, so remember this. Okay, cumulus oophorus, corona radiata, better explained on this part. So this is already your tertiary follicle, because you can see that antrum is way huge, right? So again, secondary follicle, notice how tiny the antrum is. If antrum is this big, then it is already tertiary follicle. So already you see what? Granulosa cells are still surrounding your antrum, right? You can see inside there, you have the primary oocyte. Primary oocyte surrounded by zona pellucida. And these particular granulosa cells, which are on the top of zona pellucida, they are known as the corona radiata. But they will be known as corona radiata after the ovulation, only after when the follicle ruptures and this entire structure is flying towards fallopian tube, only then this layer will be called corona radiata. So in your SCT, you will see the same. After ovulation, this area will be called corona radiata. Corona radiata consists of granulosa cells, which are on the top of the zona pellucida. Here you can see a little tiny bridge that is kind of connecting your oocyte to the rest of the granulosa cells. This is called cumulus oophorus, right? And it's just like a bridge. And this bridge is very important because when ovulation happens, it will break. Okay, so this entire area right here will be broken down. Outside, you can see theca folliculi, but already it has two layers, theca interna and theca externa. So this is at this stage, you already see the difference. And lastly, we reach the Graafian follicle. Graafian follicle is your tertiary mature follicle, but it is approximately 25 millimeters. So how many is it? It's about 2.5 centimeters. It's a huge follicle. It's visible with the naked eye. Graafian follicle is the follicle right before ovulation. So you call it Graafian approximately 24 or 48 hours before ovulation. In the Graafian follicle, you don't have primary oocyte. You have secondary oocyte because 12 to 24 hours, they say here after the LH surge in histo, but in embryo, they say before the LH surge. So in embryo, it's before the LH surge. You have the secondary oocyte formation. So histo and embryo kind of disregard on this part, but that's the most important thing, right? So before the LH surge, before the ovulation, 12 to 24 hours, the body here will be not the primary oocyte, it will be secondary oocyte already. Structures are the same. You'll see that it's exactly the same thing. Okay, so which follicle is this?
Secondary?
So
Um,
it's probably primary. Why is it primary?
There's no clear answer.
There's no antrum, exactly. So, and here the epithelium is simple. It's just the first layer, right? So it's just the first stage. What about this one?
There is an antrum, so it's secondary.
It's secondary. This one is definitely secondary. So because there is the antrum present here. Okay, the entire process of ovulation, we will talk about it next class. So we'll go back to this part and there is a area where I will draw it for you because we don't have time right now. So next class, we'll talk about ovulation. So I'm skipping this and we go a bit forward. Okay, so we go for the corpus luteum. Do you guys know what is corpus luteum?
The remnant of the
follicular.
Exactly. So after the Graafian follicle ruptures, we call it ovulation, right? So ovulation is when the Graafian follicle ruptures. So what is left behind after ovulation? What is left behind? Well, we call it corpus luteum. So Graafian follicle after ovulation becomes corpus luteum, and corpus luteum consists of two things. It consists of theca interna and granulosa cells, but they have already differentiated. They become theca lutein cells and granulosa lutein cells. Why? Because they're super, super high LH. Why is there super high LH?
the LH surge, which caused ovulation.
Exactly, because LH surge causes ovulation. So you have so much available LH. So this LH forces the remnant cells of this follicle to become theca interna lutein cells and granulosa lutein cells. This process is called luteinization. So again, super high presence of LH converts the remnant cells, theca interna and granulosa cells, into lutein cells. So what do lutein cells do? They use LH, this abundant high LH, to make two hormones. These two hormones are called progesterone and estrogen. Which one do they make it more? Progesterone. Okay, so these two cells, they will be making lots and lots of progesterone. So job of the corpus luteum is to make progesterone. Does it make sense? There is something called corpus hemorrhagicum, and we rarely see it. Basically, there are lots of blood vessels here. So when the Graafian follicle ruptures, sometimes you see that blood starts to clot in this area. So technically, right after the Graafian follicle, the next body that you have is corpus hemorrhagicum. And corpus hemorrhagicum will become corpus luteum. Because again, when you have a graphene follicle and it ruptures, it causes some of the blood to flow here. That creates corpus hemorrhagicum. And then this blood gets cleared away and you make lutein cells and you have corpus luteum. Okay, so we talked about them. Size could be important. And remember, what are they making, right? So they're making estrogen and progesterone. About 75% of their secretion is actually progesterone. Now, if fertilization does not occur, okay, so no fertilization is happening, so what will it become? What will corpus luteum become? It will become a scar. It will become corpus albicans, right? So if there is no fertilization, eventually the LH surge comes to an end, right? And your lutein cells here in the corpus luteum, they need high LH to kind of sustain, to make sure that they can live. But if there is no fertilization, LH is going down, embryo is not making HCG because there is no embryo. If there is no HCG, that means that corpus luteum cannot live. It needs HCG in order to survive. And if there is no HCG, it will die. And how do you call dead corpus luteum? You call it corpus albicans. It's an actual scar. Okay, so remember, it needs HCG. Okay, let's quickly talk about it. So here's your ovary, right? So this is the cortex and this is the medulla of the ovary. This is the meso-ovary. So this is the part of the ligament, the broad ligament, right? So this is the meso-ovary. Inside of the medulla, what can you see?
It's a lymphatic system or a vein.
Lymphatic, blood vessels, nerve fibers, okay? So basically everything else but the follicles. And this is the cortex, and you can clearly see that there are some follicles here. These huge follicles are called atretic follicles, okay? So these ones are atretic follicles. What does atretic follicle means? It means it's a dead follicle. You remember how many follicles do we choose monthly?
We choose from 15 to 20. And
15 to 20. How many will mature?
One.
One. Only one of them will mature. So what will happen to the rest of the 14? They will become corpus atreticum. They will become this part. This is a dead follicle. It's the one that didn't reach the mature stage. They didn't mature to the tertiary stage, right? So this is the corpus atreticum. Eventually, corpus atreticum will become something like this. And this is your corpus fibrosum. You can see that eventually it kind of collapses and becomes a body of fibers. So here, this structure, this is a corpus fibrosum. So this cystic corpus atreticum should go down, kind of break down and become corpus fibrosum that you can see here. This is a scar from a previous ovulation. So this is
corpus fibrosum.
This is corpus
albicans.
Albicans. This is a corpus albicans. What, Jordan?
Yeah, I said albicans.
So this is going to be the corpus albicans. So this is, how to say, this is the graphene follicle from last ovulation, like last month, right? Nothing has happened, so it became a scar. So this is the corpus albicans. And this is the follicle that have just ovulated. So how do you call it?
Well, the second term of the
Corpus luteum. Okay, so this is going to be the corpus luteum. So ovulation has just happened. So this is a corpus luteum. Okay, we're in the cortex. You can see some kind of structures like this.
Primordial.
So primordial follicle, very good. So this is the nucleus of the primary oocyte. This is the cytoplasm of the primary oocyte surrounded by the flat follicular cells. So this is a primordial follicle. How do you call all of this? This is stroma. This is stroma. And notice that stroma has lots and lots of smooth muscle cells. So these elongated cells that you have here, these are smooth muscle cells. Okay, so lots and lots of smooth muscle cells. Okay, so here we have some kind of epithelium. In our picture, it looks squamous, but this is still germinal epithelium. So don't forget, on the surface of the ovary, we have a simple cuboidal, in some places squamous, like here. Okay, in some places squamous germinal epithelium. Right under it, you can see tunica albuginea. So this black line represents the tunica albuginea. So this area here, this is the tunica albuginea. Stroma, and then you can see something like, okay, let me get you the easy one, like this one.
Primary.
Primordial, because the follicular
Primordial.
cells are squamous. What about this one? This is primary follicle
Primary.
because the follicular cells are cuboidal.
Cuboidal.
Okay. And this one?
Does this have an antrum?
No, this is an
Primordial
artifact. This is an
follicle?
artifact. This is a primordial, but it's already becoming primary, because do you see here we have the squamous follicular cells, but here they're already becoming cuboidal. So this is a primordial follicle becoming primary. This is an artifact. It's not an antrum. Okay, this one.
Primary.
Primary follicle, you can see that this is the nucleus, this is the cytoplasm of the primary oocyte. These are the cuboidal follicular cells. What is this line? Zona pellucida, very good. So this is the beginning of the zona pellucida. So it starts, it's very, very thin yet, so it's not even considered to be zona pellucida, but still, okay, it could be considered. Technically could be, it technically couldn't, because it's too thin, but you would definitely say that this is zona pellucida. Okay, again, these ones are
Primordial.
primordial follicles, all of them here, right? So all of them here are primordial. This one?
Primary.
Primary. And what do you think of this one?
Primordial becoming primary.
Are you sure?
It has an antrum.
Cuboidal, yes. And it looks like it's starting to have an antrum. So this is primary becoming secondary. It's still a bit small, but it's primary becoming secondary, definitely. What is this? Again, germinal epithelium. Yes, yes, yes. This is a germinal epithelium, right? Surface epithelium, this one.
Tunica
albuginea, very good. This one?
Stroma.
Stroma, with lots and lots of smooth muscle cells. Okay, so here we have a very interesting follicle. You don't see the oocyte. Okay, it was, I don't know, something has happened to it, maybe dissectioning or something like that, but still it's a follicle. Which kind of follicle is it?
Secondary. I will try.
Very good, secondary, because you can see that there is an antrum, but it's still a bit small. So this is an antrum, where oocyte should have been, but we don't see it. It's okay. What about these cells? Which are outside of the antrum? Which cells are these?
Corpus luteum. Well, not exactly, because
not even close. Not
no.
exactly, but not even close. Which cells are outside of the antrum, guys?
Granulosa cells.
Granulosa cells. So these are the stratified cuboidal follicular cells. These are your granulosa cells. What about these ones?
These are
they have LH receptors.
Theca interna.
Theca interna cells. And what about this one?
Theca externa.
Theca externa. Together, theca interna and theca externa are known as
theca folliculi.
Theca folliculi, very good. Okay. Take a look where we are. We are in one of those corpus atreticum, but probably this corpus atreticum was actually tertiary follicle, because do you see how huge the antrum is, right? So it was super, super big, but then ovulation happened in another one. So what are these? This is the antrum. Granulosa cells, this one.
Interna
and
externa.
Theca externa. Very important to remember that inside of the theca interna, you have lots and lots of blood vessels. Okay, so it's highly vascular area here because LH has to reach somehow, right? And that's through the blood vessels. Now, this is the corpus fibrosum. So all of this cystic corpus atreticum, eventually they will become something like this. And this is the corpus fibrosum. Right, so if you choose a follicle, you choose 15 to 20 of them, only one of them will mature. The rest of them, the rest of the chosen ones will first become corpus atreticum, like these cystic structures, and then this corpus atreticum will become corpus fibrosum. Will corpus fibrosum leave a scar? No. Okay, so this tissue will be dissolved, more stroma will be added here. It's like it was never there. Which one leaves a scar? Dead corpus luteum. So corpus albicans, it's an actual scar. Okay, you will actually see previous corpus albicans. Okay, the same idea, so you know, right? So this is the primordial follicle. This is the second stage of the primary follicle, because you start to see that these follicular cells are becoming stratified. So this is still cytoplasm of the oocyte. Nucleus is not visible. So that's what I'm talking about. Here we have corpus luteum, and this is corpus albicans. So see the difference, right? So notice the difference. So this is the corpus luteum, freshly ovulated tertiary graphene follicle, and this is the corpus albicans from a previous one. So from a previous ovulation. So this is still another slide. This is huge, huge corpus luteum, super fresh. Like literally the moment it ovulated, they got a section of this rat's ovary. Okay, so this is the corpus luteum. You magnify and this is what you see. So take a look where we are. This is the lumen of the follicle, right? So that's the area where we used to have the antrum with the liquor, with the secondary oocyte surrounded by the zona pellucida, and then later corona radiata. So how do you call the cells which are facing this antrum? So take a look where we are.
Granulosa.
Which cells are these? Yes, but don't forget that this is corpus luteum. So what is their technical name?
Lutein, granulosa.
Yes, granulosa lutein cells. That would be the full name. Okay, so granulosa lutein cells. Which cells are here?
Theca lutein.
Theca interna lutein cells. Very good. Theca interna lutein cells. Notice there is no theca externa outside. Why there is no theca externa, we'll talk about it next class when we do the actual ovulation as a process. As you can see, we're skipping a bit ahead. We didn't do the whole process of how do you ovulate, but we'll talk about it next class. What do these interna and granulosa cells do? What do these lutein cells do?
Secrete progesterone.
They make progesterone. Do they only make progesterone?
Estrogen also.
Also, they make estrogen. Okay, so very good. They also make estrogen. Don't forget about that. Okay, so here we have a tertiary nice follicle visible. Nice tertiary follicle. So what is this? What is the most outer?
Externa.
Theca externa. This one?
Interna.
Theca interna. This one?
Granulosa cell.
Granulosa cells. What is this?
Antrum.
Antrum. What is inside of the antrum?
The fluid.
Liquor. Okay, so this area, this is liquor. This is liquor. Liquor is very, very rich in hyaluronic acid. This is actually hyaluronic acid floating here with a bit of gags. Okay, so these are the proteins like hyaluronans, right? So that you can see here. Okay, inside you can see here some kind of oocyte. Notice we cannot see a nucleus due to a sectioning, okay? Because section was like this, so nucleus is not part of this picture. Does it mean that it's not there? Of course not. So let's magnify a bit into this area. So how would you call this area? Like a bridge. This is the cumulus oophorus. So like a bridge between the rest of the granulosa cells, right? So here we have granulosa cells, all of them, and the oocyte. This is the cumulus oophorus. This is the cytoplasm of your oocyte. What is this red line?
Zona pellucida.
Zona pellucida. what could this become after ovulation?
Corona radiata.
Corona radiata. So if I point here and tell you this is corona radiata, correct?
No.
No.
No, because your follicle is intact. Only after ovulation would you point at somewhere there and would say that this is the corona radiata. What is this?
Thicker internal.
Theca interna, very good. And what is this?
Thicker
Thicker
external.
external.
Theca externa. Here we have granulosa cells, right? Okay, that's it for today, guys. So we'll talk about ovulation cycle and then fertilization on all of this stuff next class. If I could ask you, please refresh your embryo one, because next class we'll talk about embryo, we'll talk about placenta, we'll talk about all of those things. I cannot teach you embryo one in this class. Okay, so this is